It's my pleasure to give you an update on where we stand with MADI Therapeutics. We're developing the Evolute, which is a punctal plug-based delivery system to deliver drugs to the surface of the eye. We've developed a proprietary device, and since we're dealing with punctal plugs, the first question we're often asked is, what are retention rates? So this is data from 2 multi-center US studies looking at retention rates over a 12-week period of time on about 180 subjects. And we average about a 94% retention rate over that period. So then the question becomes, how broadly can this technology be used? So we've studied this clinically in glaucoma. We've also studied it clinically in allergy. I'm going to present data today that's not been presented before with an anti-inflammatory, specifically napafinac for post-cataract inflammation. And we're also developing a formulation of cyclosporin in dry eye, which should show synergy given the punctal plug use for drug delivery. So let's talk about the Evolute data in napafinac. This is a cataract study looking at pain and inflammation following cataract study. It's double masked. All patients are receiving a punctal plug. There's an active ingredient or placebo, true placebo. There's a 2-to-1 randomization in favor of the active overall. Endpoints are commonly used pain and inflammation endpoints. Important to highlight that the plugs are inserted 1 to 2 days preoperatively, so they're there at the start of the procedure and carried on through. And the data I'm presenting to you today is an interim analysis of the first 44 subjects enrolled in the study, again with a 2-to-1 randomization. Baseline demographics were balanced for age, gender, and race, and the surgical procedure for cataract grade, incision size, and cortex removal. Important to highlight that the plug retention rates were excellent thus far. Only 1 plug was dislodged out of a total of 212 total patient visits thus far. And again, I'll remind you, these were placed preoperatively, so it was placed with the speculum that was used for the cataract procedure. So let's talk about data. Our first efficacy slide is patients with no pain at the day 3 post-op visit, and it's 66% in the napafinac arm versus 25% in the placebo arm. This is patients with no pain throughout the entire 2-week follow-up visits, and so that's 43% in the napafinac arm versus 0% in the placebo arm. The third pain measurement is none or trace pain at the day 3 post-op visit at 76% in the napafinac arm and 42% with the placebo arm. Turning to inflammation, this is anterior chamber cells, either none or trace. At the day 3 post-op visit, it's 86% with napafinac versus 50% with placebo. And turning to flare, again, none or trace at day 3 is 95% with napafinac versus 67% for placebo. So we're seeing consistent 30 to 40 percentage point differences between active and placebo through the outcome measurements that I've just shown you. So why are we seeing results like that? Well, I haven't mentioned it yet, but we can engineer our elution profile depending on the disease we're trying to treat. So for an anti-inflammatory, an acute condition, we actually want to engineer a high burst. So when we did that elution profile, and then we looked at the active metabolite of napafinac, amphinac, and we measured that in aqueous humor, we found these concentrations. So amphinac is a very potent molecule. The IC50 is down around 1 nanomolar. So the takeaway from this slide is during the first week of therapy, you're getting anywhere from 15 to 25 times greater drug load than you need to show efficacy. And in week 2 and beyond, you're seeing anywhere from 10 to 15 times greater drug load. And we think that's what's contributing to the efficacy results we've shown thus far. So this is really where the rubber meets the road. This is uncorrected visual acuity of 20/50 or better. At the day 3 post-op visit, it was 71% in the napafinac arm versus 50% for placebo. And at day 7, it was 83% versus 50% for placebo. So we're seeing dramatic decreases in pain, dramatic increases in inflammation, and better visual acuity with this treatment. Side effects, there have been 11 adverse events reported so far. They're balanced between the active and placebo arms at about 25%. Thus far, nothing serious or unexpected to report. In terms of the pipeline, we plan on finishing this phase 2 study before the end of this year. The next study to be started is a travoprost study in patients with ocular hypertension and glaucoma. Then we'll also be looking at a steroid, again, post-cataract, and given the results we've seen with the non-steroidal, we have a high degree of confidence that we can deliver a steroid and show equal or better efficacy. And then we'll also be completing our cyclosporin formulation And then finally, next year, we'll be moving the Napafinac program into phase 3. This platform is very well protected. The slide's already out of date. We already have 114 global patents that have issued. We're on a run rate of getting a patent issued about every 2 weeks with this platform delivery. So in summary, there's a wide range of compounds that we've already formulated. There's many more that we'd like to do. We can engineer a flexible drug delivery profile, engineering a burst or a steady state elution. This is truly non-invasive. We don't have to overcome the tear turnover issue, so we don't have to give large quantities of drug like you do with eye drops. Our device is comfortable for patients, yet we have excellent retention rates. It's cosmetically invisible, yet it's easy for the patient to identify. This is truly a preservative-free system where we put control of compliance back in the hands of physicians. There's also a favorable reimbursement opportunity and strong IP protection. So thank you very much for your time this morning.